Thank you. Good afternoon. I am Olav Hellebo. I'd like to give you a short overview of Renuron. And yes, we are unique, just like everyone else. So first to the disclaimer. They're getting longer and longer. I think we're winning the coveted prize of the longest disclaimer today. So Renuron is a clinical stage stem cell company. We're a public company in the UK. We have developed allogenic stem cells using a highly scalable technology. We have started a phase 2 clinical trial in stroke and a phase 1 trial in CLI. We're also in the final preclinical stages for a cell line targeting ophthalmic diseases. But what really gets our CSO, John Sinden, out of bed in the morning is actually his research into exosomes. Capricorn Therapeutics was mentioning exosomes earlier today, so there seems there are more people that are excited about it. That's great to see. Exosome is a field of vesicular transfer of cellular information, and it is an exciting new emerging area. So we're delighted to be part of that. Also want to mention that we have a very strong investor backing, particularly in the UK. And we're in the process of building a first-class cell manufacturing facility in Wales, and that's backed by a government grant. Our technology platform consists of 2 cell lines, and they're being investigated in 3 different indications. The CTX cell line was derived from brain tissue in 2002. A single neural stem cell was selected, conditionally immortalized and cloned. So these CTX cells are currently in clinical trials in 2 indications, and we're also extracting exosomes during the CTX manufacturing. The retinal cell line, human retinal progenitor cells, are derived from fetal retinal tissue. So it's not a clonal cell line, but a more conventional cell population expansion. We're using a patented low oxygen culture to do that, and this technology has been licensed from Dr. Young's lab at Harvard. So since the CTX cell line is derived from a single cell, it is easier to deliver consistent output from manufacturing. I mentioned that the manufacturing is scalable and cost-efficient, particularly at high scale. Last year, we were able to develop a cryopreserved CTX drug product, and this was a real breakthrough for us. It has improved the shelf life significantly and also been very well received by our clinical sites. Exosomes. So some years ago, it was thought that cells were more or less islands with signaling limited to physical contact between cells or chemical release and uptake. This understanding has now changed. There was actually a Nobel Prize in this field last year for Sheckman, Rotman, and Sudoff. So very, very interesting emerging area. There's extensive protein and genetic information sharing between cells, and exosomes are thought to be a very vital part of this. The exosomes are nano-sized vehicles secreted by stem cells. They contain multitudes of RNAs, cytokines, and other proteins. Exosomes so far have been used as diagnostic tools, and we are now investigating their therapeutic potential across various indications such as wound healing, immunology, and cancer. So this is our pipeline in the snapshot. So we are in phase 2 for stroke. This trial is ongoing across 9 sites in the UK. We've started a phase 1 study in CLI with the same CTX cell line as in stroke. We're conducting preclinical studies in retinitis pigmentosa with our human retinal progenitor cells. And as mentioned, we are looking at our CTX-derived exosomes across several indications preclinically. So let's have a closer look at the stroke program. In the US alone, there are 800,000 stroke victims per year. Their outcomes range from death to spontaneous recovery, About a third will be left with chronic consequences such as paralysis, and this has an obvious large social and economic consequence. The pharmaceutical treatment window is currently only during the first few hours post-stroke, and hopefully Athersys will enter that area with Multistem when we're waiting for the data to come there. But after these first few hours post-stroke, there is no therapeutic intervention. It really then ends up being physical rehab that's being used. So our aim is to change that and improve recovery in chronically disabled stroke survivors. Preclinical models have shown that our CTX stem cells induce growth of new neurons and blood vessels, as well as triggering the immune system to reduce inflammation. So let's look at the clinical data that we have so far. Phase 1 dose escalation study was performed in 11 patients. The patients suffered from moderate to severe ischemic stroke, and the treatment was performed an average of 2 years after stroke. It's a long time. The stem cells were delivered via standard stereotactic surgical procedure, and patients spent 1 or 2 nights in hospital. No adverse events were attributed to the stem cells, nor were there any immunological AEs. While this was a safety study, we obviously did look at the range of efficacy measures, and there was a consistent improvement across these different efficacy scales at 3 and 12 months. So that encouraged us to go into a phase 2 study. But we have made some changes between phase 1 and phase 2, and I think these are great improvements. First of all, we're treating the patients much closer to the stroke episode than we did in phase 1. We believe that the ideal treatment window is after the motor deficit has become stable, and that's about 30 days, but before significant scarring has occurred. So we're enrolling patients 1 to 2 months post-stroke. We observe them for a month to make sure that they're stable, and then we treat them. So rather than 2 years post-stroke, we are now treating 2 to 3 months post-stroke. Over to CLI. CLI leads to a surprisingly, at least to me, large number of amputations every year, most of them in diabetics. Any treatment that can increase the blood flow in the lower limbs to reduce this number would be a great medical advance. Benefits have been shown using autologous stem cells in CLI already. However, there's been some issues around the quality of the cells and also the time to produce the cells in order to benefit the patients. And we're seeing an opportunity here to build on what's been done already and improve on it with an off-the-shelf approach. So we have kicked off a phase 1 safety study in 9 patients in the UK. We expect the results in the first half of next year, and if there are no surprises, we will start a phase 2 study by the end of next year. Now, our third program. Retinitis pigmentosa is a degenerative eye disease that can lead to blindness. So we have developed a human retinal progenitor cells that we have tested in various animal models. The preclinical data shows a prevention of loss of visual acuity in the RCS rat. We also see allogenic engraftment as cells migrate into the photoreceptor layer in pigs' eyes. So this is exciting. We may both protect and replace photoreceptor cells. We're finalizing this work now, and we will file for an IND early 2015. So this is what we aim to deliver. If you start from the bottom of the chart, stroke, phase 2 has started, with data expected next year. CLI is the next line above. Phase 1 has started, and we will start phase 2 next year. The retinal program, IND in early 2015 for a clinical start by the end of the year, 2015. And the top line there, or second top line is, top line is exosomes. We plan to get one of our indications, and we haven't selected one yet, into the clinic in 2016. So in summary, our allogenic technology platform consists of 2 cell lines being investigated in 3 different indications. We're well backed and well funded. We raised $40 million in equity last year, as well as $20 million in grants. So we have the funding in order to get in goal with the different milestones that I just showed you. We are very excited about the clinical advances that we made so far, as well as our research into exosomes. Thank you very much for your attention.